I'm trying to join the meeting again. I don't know what do I have to
Okay.
do?
Good morning, Enid.
Good morning.
Is it as cold in New Haven as it is here, Noah? Oh, here comes Mark Levine.
How
Yay.
cold is it in Hamilton?
I think it's around -12 or something like that.
As I walked to work this morning, I'm coming to you from work.
You're at the lab?
I'm across from the lab. We could walk to the lab. I could show you if you want. But no, it was -10 and windy. It was very
Oh,
chilly.
very cold there too, eh? Wow.
Yeah.
Okay.
Two people entered the waiting room.
Oh, Jacqueline's coming and Penny again. I got them.
You got them. Okay,
Good
great.
morning, everyone. Here comes Ginny, and she was with us last night at trivia. And I split them up. So Ginny was on one team, and her husband Mitch was on the other team. And Mitch's team won. It was a date night gift. It was a giant Toblerone and a bottle of wine. And so Mitch's team won, and they split it up. Mitch took the bottle of wine. So Ginny won either way, took the bottle of wine home. And I think Jax and Sam took the giant Toblerone. And that's how trivia went down last night at Temple on Shea Shalom. Hi, Mark. Now we can start because Mark is here, but we should wait 2 minutes in case anyone else wants to come. Jacqueline, so good to see you from St. Louis. Love it.
2 weeks in a row, Jacqueline.
Yes, you're becoming a regular at Breakfast Club.
Jacqueline's in my slideshow.
Is she?
Just one picture.
Okay. Oh, here comes Erica Appel, our junior scientist to be. Erica.
Hello.
Hi, Erica. Thanks for coming.
Thanks for doing it.
You're truly next generation.
Sorry,
And
I'm walking to school. That's why my camera's not on, but I'm paying attention, I swear.
Both Noah and Jacqueline are here online, as well as Dr. Mark Levine. I know is instrumental in you connecting to what you're doing at MAC, which is so.
That's so true.
Oh, right.
So your visas haven't been tacked yet,
No, we'll see. We'll see what
or
happens.
your NIH funding. But anyways, let's talk about good things. Yeah.
And funding, not good.
I was surprised to learn, although we did just submit an NIH grant, or I should say my boss did, that our lab does not have and perhaps has never been NIH funded, despite being extremely relevant to health and infectious disease research. We have a lot of private money, which is good nowadays.
Mm-hmm.
Oh, you can. The Hinda's just arrived, so it's now cool.
So and we got just watching the clock on my computer here, assuming that is accurate. 10:59. So being patient. Good morning, Hinda. Can she hear me?
Good morning. Yes. Hi.
Yes. Hello. I know you've got the
Hello.
coffee on. You ready to go? And here we are at 11 a.m. And it's my pleasure this Sunday at Breakfast Club, to introduce you to my baby. Yes, Noah, said it.
Yes.
Noah Houpt, the youngest of my children. And I just lost your bio, Noah. I'm going to let you.
I'm going to introduce myself.
It's terrible. Thank you for being with us, Noah. We had your beloved Jacqueline last week, and Sam Bader was our first presenter. And in 2 Sundays from now, right after Family Day weekend, we have Rhonda DeHaan, which will conclude our next generation for this year. And our next Breakfast Club will be in with different presenters, including Dr. Mark Levine. Will be in April. My goodness, fire engines are going past outside. Yes. We're going to get started, and I'm going to turn the microphone over to Noah Houpt, who's in New Haven at Yale University. Noah.
Rhonda is going to present, or Renan?
Renan.
Okay, Renan.
Yeah.
Yeah, okay. That'd be a good one for Rhonda. And I think that you've already had a sibling of mine maybe present. Is that true that I'm the second of your children? You had Rebecca?
Well, Daniel, I'm going to count him as a sibling of yours, even though he's married in. And Rebecca and Jax presented in a teacher's segment. Okay,
so I think I'm fourth then.
Yeah.
Low priority perhaps, but hopefully I'll be among.
I think I recall in your first year at Yale, I asked you and you said, Mom, I'm not ready.
I don't know what I'm doing. That's still true, but maybe I don't care as much.
Okay, there you go.
I can get going. I suppose I should ask, can people see my slides?
No.
Oh, you can't. Oh, because I got kicked out and I need to reshare. Let me try it again.
Are you still a
I
co-host? Yes.
think so. I think I am. How about now?
Yes, I see them.
Terrific. Okay, we can get started.
Yay.
Okay, well, thank you for having me, Mom/Cantor Baruch. I think I'm going to address you as Mom, but we'll see how it goes throughout. And thank you everybody for joining. Today I'm going to talk a little bit about my scientific journey, my journey within academia, and generally this philosophy that I wanted to share with you guys about evolutionary thinking and what that means to me. And as I'll get into a little bit, it means trying to approach biological problems from an evolutionary perspective, which is just one of many perspectives and approaches we can take to solve problems, as I'll get into a little bit later. But first, since my mom didn't have the intro already, so let's ask the question of who is Noah Houpt and what's going on here. I saw this on Instagram, which I'm pretty sure my mom makes these posts on Instagram. And according to Instagram, I'm a biotech medical researcher. Not quite accurate, which I think you'll be aware of by the end of this talk. I'm just bullying my mom a little bit. It's all good. But let's try to figure out a few different dimensions of who is Noah Haupt. So, as my mom said, Noah Haupt is the son of Cantor Paula Baruch. Here's us in New Haven at trivia, actually. Speaking of trivia, and here's me. This is a GIF that my brother Sam made of me right outside the front of the temple, which I don't know if you can tell that that's right in front of the temple, and TD is somewhere around here, around the corner here. It's one of my favorite little videos that Sam's made. But you probably care for this breakfast talk, since I'm going to be presenting about my scientific journey, about maybe some of my background in research and education. So I did my undergrad just like Jacqueline, who we heard from last week, at McMaster University in the Integrated Science program, an interesting little small program that focuses on, tackling big scientific problems from multiple perspectives. In my final 2 years, I worked with someone who some of you might be familiar with, Professor Sigal Balshine. I worked in her lab on studying the behavior, physiology, Michelle, and ecology of different fishes. And I did my honors thesis with her, which I'll talk about a little bit more. And I will heap more praise on Sigal a little bit more later. That's my oceans segment, by the way. And after working with Sigal for undergrad, I did my Master's of Science in Biology at the University of Ottawa. This is during the pandemic, the worst of the pandemic. And I worked with this guy, Professor Reese Casson, who, by the way, I don't know if my mom knows this, but went to like some sort of Jewish summer camp in British Columbia with Sigal. And so they knew each other, which was quite a surprise to me when they started sort of communicating back and forth about this guy, Noah, and whether I should be, admitted into the Casson lab. This is my transition from working on fishes to working with microbes, and I'll talk a little bit later about why I made that transition and why microbes are such a fascinating system for understanding evolution, and they're important in their own right. Let's not forget that. And then a little bit about what I'm doing now. So since 2021, I've been doing my PhD in ecology and evolutionary biology here in New Haven at Yale University. I've been working under the supervision of Professor Paul Turner, who's an amazing, brilliant guy who spends most of his time these days, as I'll talk to you about in maybe 30 minutes, trying to figure out how we can use viruses to solve the antibiotic resistance crisis in bacteria. Ooh, we'll hear about that a little bit later. Before I go into evolutionary biology and sort of the research that I've done, I wanted to just acknowledge the people who have helped me along the way. And as promised, heaping praise on Sigal. Here's Sigal at Lake Tanganyika in is sort of a dry suit to go scuba diving and study fishes there, cichlid fishes. I just wanted to specifically highlight Sigal. No shade on my other advisors that I've had throughout the years and mentors, but Sigal really is, as I'm sure some of you know her and you might know a little bit about her research, but what you probably don't know is she's an exceptional mentor, especially for undergraduate students. Both Jacqueline and I were in her lab, ABLE, Aquatic Behavioral Ecology Lab, at McMaster, and the amount of effort and focus she puts in to bring along undergrad students and reveal to us some things that are sometimes called the hidden curriculum of science, which is stuff like, how do you make a scientific paper? How do you reach out to another researcher to start a collaboration? How do you present your work? And she even gives undergraduates the ability to lead scientific projects in some cases, which I was lucky enough to do. And that has really set me up for a huge deal of success. So Sigal really is, I've been in several places now, I can say this with confidence, an exceptional supervisor, especially of undergraduate students. So I have to thank Sigal and heap praise on her lab for all the help throughout the years. Here's my picture with me and Jacqueline. Jacqueline's also been extremely helpful and throughout my scientific journey, as has my family. My family's also been very helpful. The members of the Casson Lab, especially Sunal Shiromani, Angela Alonso Morales, and Parto Chakraborty. And here at Yale, people like Kathy Hernandez, Helen Stone, Dallas Mold, Asher Leaks, and others, Katie Courtwright, not pictured, have been so helpful. So a lot of the stuff that I've produced and the things I know comes from working with them. Okay, now I can get into the scientific content of what I want to talk about. So the first concept I want to talk about is, what is biological evolution? Why do I say biological? Because it's often conflated with the colloquial term evolution that we just use sort of in our day-to-day lives, which usually means progression or getting better. Biological evolution is something different. It's something specific, and it refers to changes in living things over generation. It doesn't necessarily mean getting better. Just wanted to emphasize that. And this is, it's a pretty significant concept societally, but I'm mostly going to focus on the scientific side of things. So biological evolution is the change in living things over generations. We can understand what this change might look like by looking at some classic examples in evolution. Some of you may have heard of these little critters on the left here, which are Darwin's finches. Darwin's finches are pretty special because, Darwin, when he was surveying the Galapagos Islands as part of this big, long, global, nearly research trip that he took, he noticed these birds, which were extremely closely related and generally similar. But as you can see in this picture, they had really different beaks. And those beaks allowed those birds to sort of specialize on different food sources in their environments. And he wrote something that I think is pretty striking because he didn't really know what he was talking about at the time. It was before he came up with the idea, published his various ideas about evolution. And he observed, in reference to these birds, he said, seeing this gradation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds, meaning from originally one species of birds or one population of birds, from an original paucity of birds in this archipelago, these islands, one species has been taken and modified for different ends. So he made that as kind of a hypothesis. Maybe from an original population of birds that all look similar, they've changed over time to fill these different niches in their environment. They've changed their beak dimensions in order to specialize on different foods. That's biological evolution, change in living things over generations. Another example that I won't dwell on too long, but you should Google it if you're interested. It's pretty similar to the Darwin's finches stories, but I would say more striking. These are the Anolis lizards, and they have colonized the various islands of the Greater Antilles. So this is Cuba and Puerto Rico and Hispaniola and Jamaica. And each time they've colonized these islands, they've also changed. And they've changed by splitting into different species, just like Darwin's finches, that are specialized on inhabiting different parts of the trees. And that's led them to be really different in body size, really different in limb dimensions, stuff like that. So it's like an even more striking, if you will. Here, let me admit, Margie. Really striking example, similar to Darwin's finches. We can also see change in living organisms through unexpected marvels. I don't know about you guys, but growing up, I saw whales and I said, well, those are surely fishes, right? Those look like fish. They look like sharks, stuff like that. But in reality, when you look at the evolutionary history of whales, when you look back in time, the progenitor of whales was actually a quadrupedal, a four-legged land mammal, similar to, oh, Penny's back. Similar to rodents, you might look this and see it looks like a rodent. This one happens to be underwater, and part of the transition to being more like a whale was a land mammal starting to spend more time in the water. But over millions of years of small incremental changes, you go from a quadrupedal land mammal into this gargantuan of the ocean that spends all of its time in the ocean. And if you look at the skeleton of a whale, you can see things that you'll never see in a fish. For example, you'll see the vestigial remnant of hip bones, which fishes don't have. But it's sort of this calling card, this scar, if you will, evolutionary scar of, the evolutionary history of being a land mammal. Okay, so these are both really deep time, long sort of processes that created these patterns. But what about this? This chart is showing evolution, change in an organism over time. It just happens to be an organism, a virus in SARS-CoV-2 that caused a pandemic and whose evolution was crucial to the longevity of that pandemic and how it kept rising and falling over time. So if you just focus on this top part of the chart, this is like the pre-variant of concern time where we're going through that initial wave of COVID. And then you might remember, oh, now we need to worry about the alpha variant. We need to worry about that. It's more transmissible. It's more virulent. Then the Delta variant came along, and that was a lot more transmissible and virulent. And that was pretty freaky. We see evolution of this virus changes over time with different mutants taking over the population. And of course, I'm sure we all remember Omicron coming out of nowhere. And within it was just a matter of a few months, going from a very small percentage of cases being Omicron to virtually every single case being Omicron. And that all has to do with changes in organisms over time. In this case, a virus responding to built up immunity in the human population because of vaccinations and prior infections, and evolving to avoid that immunity at the same time as it evolved to better infect our cells and become less virulent with Omicron. Okay, so that's evolution, the change in organisms over time. Now we can talk about what is natural selection. Natural selection is not the same thing as evolution. Natural selection is we would call it a force that can generate a particular type of evolution known as adaptation. So what is natural selection? A textbook definition would be the differential survival and reproduction of heritable types. Now, this is an extremely concise definition, and it's not very well explained, but we can break it down to try to understand what is natural selection a little bit better, because this sort of definition contains within it the different ingredients necessary for natural selection. And when we look at those ingredients, you'll see this is not some super complicated process. It's just the natural result of 3 ingredients that occur very basically universally in biological populations. So it's the differential survival and reproduction of heritable types. What do we mean by types? I mean that within a population, you need variation in some trait, right? This could be a lot of different things. In this example I'm going to use here, it means variation in the body size of fish. So within a population, we have variation. We have different types present in the population, variation in a trait. We need there to be heritability of that trait, which is to say we need the fishes that have a larger body size to have offspring that are also larger on average. And we need the smaller fish on average to give birth, to have offspring that are smaller on average. So we need variation in a trait, and we need that trait to be at least somewhat heritable. Those are the first 2 ingredients. What's the third? Well, it's that first part of our definition here, differential survival and reproduction. We need variation in that trait, so in this case, the different sizes of the fish, to correspond to variation in the survival and or reproduction of the individuals in that population. So in this case, I'm using an example of overfishing of large fish, which is common in fisheries around the world. Usually, if we catch a big fish, we say, okay, we're going to take that, we're going to use that as food. If we catch a small fish, even if it's an adult fish, we say, I don't want that, not worth my time, throw it back. And in this way, we can create differential survival of larger versus smaller fish, right? And that's what these X's represent. Okay, so we've got variation in a trait, it's heritable, and that trait is associated with more or less survival and or reproduction. Let's put it all together. What is natural selection? The differential survival and reproduction of heritable types. So now let's imagine we start with a population that's mixed, half large fish and half small fish. You could also consider a population where size varies continuously, but just for the sake of this example, let's imagine it's half large and half small fish. And then all of a sudden, we have differential survival. We have differential death of the larger fish. They're getting overfished, and the large fish are being eliminated from the population. Those fish are being replaced by the offspring of the small fish or the remaining fish in the population, which are mostly small. And then if you iterate this over a few cycles, all of a sudden we have a change in the organism over time. We've gone from a population that's, on average larger to a population that's on average smaller. And that's what natural selection is. It's the differential survival and reproduction of heritable types that drives evolution. Okay. A little bit more on natural selection. So like I mentioned, natural selection is the driving force of adaptation. Adaptation's a special type of evolution, and it's what leads organisms, living things, to have a better fit to their environment. What do I mean by fit? In this environment, large fish are killed, small fish survive. The small fish are more fit to their environment because in their environment, small fish don't die. Another example might be in an environment that's replete with antibiotics, it's filled with antibiotics that kill most bacteria. It's the bacteria that are resistant to that antibiotic that don't get killed that are most fit to that environment. We can talk more about that later. So when I say fit to their environment, this is the fit when we say fitness in evolutionary biology or when we say survival of the fittest. This is probably the most common misunderstanding of fitness or evolutionary biology that I come across. Survival of the fittest does not mean survival of the most physically fit, the largest, the strongest, the fastest. No, it's survival of those organisms that are most fit to the challenges they face in their environment. Just a few more facts about natural selection. Natural selection is why I was confused as a kid, right? And I thought whales were fishes because whales and sharks look so much alike. And that's because natural selection has led to both of these aquatic animals to have these streamlined, smooth bodies for cutting through water. They face similar challenges and they've evolved similar traits for dealing with those challenges. Like I was mentioning before with antibiotics, natural selection is also why our antibiotics are no longer effective in many cases. Because when we impose that environmental change where only the bacteria that can survive antibiotics end up surviving, then there's natural selection to be resistant to those antibiotics, and all of a sudden our drugs don't work. Natural selection, as a fun example, is also why I personally, can't speak to you guys, can drink milk without getting an upset tummy. Lucky me. And that's because certain human populations, really long time ago, started cultivating animals that produced milk, and they started drinking that milk well into adulthood, and that created a change in the environment and provided natural selection to be able to process that milk late into life. So if anyone tries to tell someone that they're weird because they're lactose intolerant, that's absolutely wrong. Our ancestors were lactose intolerant. I'm the weird one for being able to process milk. That was the change over time. Okay, so we've talked about evolution. We've talked about natural selection. Let's put those together and talk a little bit about what I call evolutionary thinking. So evolutionary thinking is about seeing biological problems through the lens of evolution. There's a lot of different ways we can tackle problems in society. Evolutionary thinking is just, let's see this biological problem and let's think about how evolution, those things we talked about before, changes in organisms over time, natural selection, adaptation, can help inform those problems. And I'm going to share with you one of my favorite examples of how evolutionary thinking helped debunk what some people would call a conspiracy theory, and has to do with the origins of the virus HIV-1, which is the main virus that causes acquired immunodeficiency syndrome, or AIDS. So as I'm sure many of you are aware, the AIDS epidemic in North America started in the 80s, or we became aware of it, I should say, in the 80s, and caused so much death and mortality. It was a huge problem, and we quickly discovered that it was caused by this virus HIV-1, human immunodeficiency virus 1, mainly. And in the 90s, there was this theory about where HIV came from. Most researchers said, well, most likely this came from a natural exposure of somebody, probably in sub-Saharan Africa, to an ape or a primate of some kind that was infected with a similar virus. The virus spilled over into humans, and then that started the global AIDS pandemic. But some people had different theories. Some people who, were skeptical about vaccines, perhaps, had this idea that, hey, maybe HIV was actually introduced into human populations as a result of trials for the oral polio vaccine. Polio, this horrible virus that's killed so many humans over generations and generations until the vaccine came into play. And the vaccines were being developed. There's a Salk vaccine, Sabin vaccine. I could talk about some of the history there. But one of the vaccines was called the oral polio vaccine, and it was developed in the late 50s, and it was tested in sub-Saharan Africa. Now, crucial to this theory that said that those tests in Africa, the theory was that those tests in Africa is what actually exposed humans to HIV. And what's crucial to this theory is about how this vaccine is made. The oral polio vaccine is made by taking polio and growing it for many, many replicate days and days and days and days in the cells of different primates. And that makes the virus that you get at the end of that not so harmful to humans, but it still allows humans to have an immune response to the real thing, the real polio virus. It's called an attenuated vaccine. Now, these people, especially this one journalist at the Rolling Stone, thought, well, hang on. Some of those primate cells might have had HIV in them. And maybe as the polio vaccine was being produced, maybe it picked up some of HIV and then we inoculated people in sub-Saharan Africa during these oral polio vaccine tests with HIV, and that's what kicked off the HIV pandemic. Now, researchers at the time were very skeptical. They took a lot of precautions to avoid that happening, but they couldn't really say definitively that that wasn't what was going on. And I just wanted to highlight this poster, by the way, from the US CDC. It's from 1963. It's encouraging people to take the oral polio vaccine, talking about all its various benefits. I would have led with prevents polio. I wouldn't have led with tastes good, but I guess that helps them, get more people to take the vaccine. It is an oral vaccine, so it means you put it on a little sugar cube and then you take it that way. Okay, so how are we going to use evolutionary thinking to sort of debunk or reveal why this theory is impossible? And remember, those vaccine trials in sub-Saharan Africa happened in the late 50s, early 60s. Okay. So along comes this paper in 2003 by Korber et al., published, I think, in Nature. And what they did was they applied evolutionary thinking to this problem to try to understand when did HIV originate in human populations. And they use this method I'm not going to talk about too much. We can talk about it in the question period. It's called the molecular clock. And what it does is they consider all the diversity of HIV, how different the different strains are from one another in the present day, or the present day at that time. They looked at all the DNA or the RNA, different genetic material, and the HIV virus, and they used a model, an evolutionary model, to project back in time, back to what they inferred would be an original transmission into a human. The whole pandemic was started from a single virus getting into a single human, and they wanted to project back how long it must have been since that happened. And their analysis really clearly revealed that HIV has been circulating in human populations and had been for decades before the oral polio vaccine trials. So they were able to use this evolutionary analysis, considering how long ago has it been since HIV was transmitted into humans, to debunk this conspiracy theory. And that's not mainly what the paper is about, but they do address how they can debunk that conspiracy theory with this analysis. They estimated HIV entered human populations around 1930, and in fact, subsequent analyses using more different strains of HIV have pushed that way back into the early 1800s as our best estimate. Okay, so that's one example of using evolutionary thinking to solve a problem, in this case, a conspiracy theory, which led some people to be really skeptical of vaccines. Now I'm going to talk a little bit about how I've used evolutionary thinking during my career. I want to make sure I get through as much of this content as possible, so I'm going to keep my eye on the time. I'm going to talk to you about just two stories. I'm going to kind of skip or breeze through this middle story because I don't have enough time. But I'm going to talk to you about some of the work I did with Segal here at McMaster, using evolutionary thinking to understand parental care in a really weird species of fish. And then at the end, I'm going to talk a little bit about how our lab uses evolutionary thinking to try to figure out the best way to use killer viruses to treat bacterial infections. Okay, the first story is about these fishes. Here's a title slide from the last time I presented it. I just snuck that in here. I last presented this actually in Hamilton at this conference that they had there. But this story is basically rooted in this question that a lot of evolutionary biologists have about parental investment or parental care. I'm sure a lot of you all are parents, and you know how much effort it takes to rear offspring, to bring them up, to make sure they're healthy, well-fed, they have time for play, all these different things. It takes a lot of investment. The same's true in animals. If these birds, these parent birds, have to go around collecting all this food more than they need to sustain themselves in order to partition food to their offspring. And if they don't do that, the offspring's going to die. If this father frog doesn't carry its tadpoles around, they're going to desiccate and have no food, and they're probably going to die. Now, what's interesting about parental investment is that it can lead to a conflict between the best interests of the parent and the best interests of the offspring. The offspring naturally wants as much investment as possible. Give me more food, give me more attention, all these different things. The offspring wants it all. The parent wants that for its offspring, for sure, but it doesn't want to kill itself providing as much effort, as much food, as much resources as possible to that offspring, because it might want the chance to reproduce again in the future. there's a conflict between what's best for the parent, which is to, yes, care for its offspring, but also continue to reproduce, compared to the offspring, which wants all the attention of its parent. And sometimes the parent has to do things that isn't optimal for itself in order to take care of its offspring. That just doesn't just include parental care after the offspring's born, but it can also include choosing where to actually rear offspring. So in this example, this is from water python in Australia. And what these researchers, Madsen and Shine, found was that these snakes can nest in a variation of different sites, ranging from colder sites to warmer sites. And they found that the snakes often choose colder sites to rear their offspring because there's fewer predators to eat their offspring. But it's actually really challenging for the snakes to exist in this cold environment. It's very energetically demanding. So there's this conflict between what's best for the parent and what's best for the offspring in terms of where to rear their young. That brings me to this guy. Okay, this is weird, right? If you wander along a rocky intertidal beach somewhere in the Pacific Northwest, you might encounter a site that looks like this. This is a plainfin midshipman who's seemingly struggling, desiccating, doesn't look too good with its razor-sharp teeth and its opercular spines here. But it looks really desiccated. It looks like it's struggling to cope with being a fish out of water. And this sort of picture begs the question of, like, how did the fish end up in this situation? And the answer comes from the unusual breeding strategy of these fish. So each year in April, midshipman males, potential fathers, migrate up from about 400 meters below sea level, so pretty deep ocean fish. They migrate up into this intertidal region, the region in between where high tide comes up and low tide goes out. And once they're there, they burrow underneath rocks and they actually sing to attract females up to their nests, who lay these beautiful orange-yellow eggs underneath on the undersurface of their rocks. And they stay there in the intertidal zone, caring for these eggs for up to 60 days. And that's all well and good. They do put in a lot of investment trying to understand, trying to care for those eggs while the water's up. But one of the big problems is that twice per day, their environment goes from looking like this to looking like that as the water goes out. And this is a big problem because it leaves them in situations like this. They're either left in a small pool of low oxygen water. They really quickly deplete the oxygen from these small pools of water they're left in. Or if they're really unlucky, which is quite common, they find themselves totally air exposed, fish out of water, like the fish we saw before. Now, this is a conflict, and I should mention the reason we think why they nest in the intertidal zone is because it has a lot of features that seem to be good for offspring development. Warmer temperatures leads to faster egg development, potentially. Oxygen, when they're air exposed, can help more oxygen actually diffuse into the eggs, which can potentially help them develop more quickly, but it seems to be pretty dang costly for the fathers that have to stay for 60 days and endure many cycles of these low tides. Okay, so like I mentioned, they're dealing with a lot of stressors here. And what we kind of wanted to understand is, can we quantify the costs that these parents are paying? We think they ought to be paying a really high cost for nesting in this extremely challenging environment. For heaven's sake, they're being a fish out of water, sometimes twice a day for 60 days. What kind of costs are they paying for this? For my thesis work, we focused on lack of oxygen. So they're found in these pools of water that have very little oxygen or completely air exposed. How are they dealing with that? What's their physiological response to that? And using sort of coming back to this idea of evolutionary thinking, we also wanted to understand what adaptations they might have. What strategies might they have for dealing with this low oxygen environment, which might reduce or mitigate the costs of oxygen stress? Okay, so these are kind of the questions that I've just mentioned here. I'll talk to you a little bit now about my experimental design. So we collected a bunch of these parental males from the field, and we kept them in the lab at the University of British Columbia. And then every day for 15 days, we would take 3 parental fish that were around the same size, and we would expose them for 6 hours to one of 3 treatments. Either normoxia, where the fish have lots of well-oxygenated, free-flowing water in their tanks. Hypoxia, where we left them in a stagnant, small amount of water, which they quickly depleted all the oxygen from. Or complete air exposure. We siphoned all the water out, and they were left without water for 6 hours. It sounds cruel, admittedly, but in reality, these fish, by all, the signs that I could take from them, were fine, and none of the fish even came close to dying during this treatment. And that's a bit of a spoiler alert for what we found in this study. But before we get to, I'll talk to you a little bit about what we measured. Once these fish underwent this 6-hour exposure of these different simulated tide conditions, this is simulating low tide, this is simulating high tide, we dissected the fish and took a bunch of their tissues, and then we measured a bunch of different molecules in those tissues, as well as a few other measures, which I'll get into in a little bit. But first, I wanted to give you a quick refresher on physiology and metabolism, just because maybe it's been a few years since you've thought about metabolism. So I want you to start here. Glucose is sort of the fundamental energy source for animals. Oftentimes we try to, if we have sucrose, for example, table sugar, or we have other types of carbs, it's often shuttled in some way to turn it into glucose. And when you have a carb-heavy meal, your blood glucose levels will spike. And a really important balance for animals is the conversion of glucose, if we're not going to use it right now, into glycogen, which is this big molecule that's a bunch of, each of these is supposed to represent a glucose molecule all chained together. That's mostly stored in the liver. As you may know, dysregulation between turning glucose into glycogen and vice versa is sort of what causes diabetes. Both types have to do with this balance between glucose and glycogen. So glycogen is sort of the long-term storage of energy in the liver. Glucose is what we're going to use right now if we need to use it in the moment. And there's two different ways that animals can metabolize glucose to make energy. If animals have lots of oxygen in their tissues, they can do what's called aerobic respiration, which is metabolism with oxygen. And that's really awesome because it's extremely efficient. It produces a lot of energy per unit glucose, per molecule of glucose. And it has pretty harmless byproducts, water and then CO2, which we just breathe out. No worries. But if there's not enough oxygen, then we have to undergo anaerobic respiration or metabolism without oxygen. And this is bad on a couple fronts. First of all, it produces less energy per unit glucose, a lot less, something like 15% as efficient. And also has this harmful byproduct of lactic acid. And that causes our tissues, wherever that type of metabolism is happening, to become more acidic. And that's sort of the pain that you might feel if you're a sprinter and you're trying to sprint as fast as you can, faster than you can even get oxygen to your tissues. Ends up with this acidic burn that you feel. Anyway, this is all to say that what we're kind of looking for in these fish, because of the lack of oxygen, is we're looking for a depletion of their glycogen stores, potentially, because they're using this less efficient type of metabolism. And also we're looking for the buildup of lactate or lactic acid in their tissues. And those would both be indications of disturbance. And if those levels were severely extreme, extremely low glycogen, extremely high lactate, that might even indicate that they're near death. So what do we find? So in these plots, these are plots from the data we collected. In blue, you see the results for normoxic fish, fish with lots of oxygen. In yellow, that's hypoxic, water with little oxygen. And in red, it's air exposure, where they had zero water, which they could breathe through with their gills. And what we found here is that for plasma, which is in their blood, and in their muscle, which we dissected out from their fin, we found that there was elevated lactate in the fish that we exposed to just total air immersion compared to the normoxic fish. So this is indicating they are showing signs of undergoing this anaerobic, costly anaerobic respiration. But kind of sussing out what this really means, we need to take more of a biological interpretation of how high is their lactate really? And in reality, taking a biological perspective of what this really means, their lactate was elevated. We can pick that up statistically, but their lactate is nowhere near a level that would be really harmful for these fish, causing lactic acidosis and really problems for their cells. You know, for example, in humans, you want lactate to be relatively low in this range, 0 to 1, 1.5. If it gets above 4, now you're kind of in trouble. And if it's sustained above 4 for a long time, that could lead to death. But even our most stressed out fish were below 4. So they're dealing with this 6 hours of air exposure. This is after 6 hours, relatively well here. That's also reflected in the glycogen results. So what this NS means is that we did not detect any significant differences in glycogen stores across these different tissues that we looked at, muscle, heart, liver, across our different treatments, our fish that were exposed for 6 hours to these different treatments. So not only do they have, sure, elevated lactate, but not too high, they're also not significantly depleting their glycogen stores. So all in all, they're not really doing too bad despite the really extreme exposure. And to sort of explain that, we wanted to look at what responses they might have, what strategies they might have to help them cope with these challenging conditions. And I'll just briefly, because I'm running long and rambling, go over one idea that we investigated and found some evidence for. This right here, I'm sorry if it's gross. This is an organ that you can find inside the fish called their swim bladder. It's not at all connected to the outside world. It's not like lungs. It's only connected through their arteries and veins inside the fish, and it's quite big for their size. And when you stick an oxygen probe inside this organ, you'll find that the oxygen levels are astronomically high. They're higher than even ambient air, and they're certainly much higher than the amount of oxygen that's dissolved in water. And so they're somehow able to concentrate a really high degree of oxygen within these swim bladders. And what we found in our experiment was the fish that had been air exposed for 6 hours actually had lower oxygen content. So what might this suggest? Well, it might mean that one of the ways they're mitigating the challenges of air exposure might be because they're using their swim bladder a little bit like an oxygen tank for a scuba diver. And they might be drawing slowly but surely oxygen stores from their swim bladder while they're air exposed and can't breathe in order to sustain a little bit of that more efficient metabolism in order to keep them, healthy and alive and able to, wake up when the water comes back in. So all in all, we found that, yeah, these midshipman males did show signs of disturbance in response to these simulated intertidal conditions, but they were able to mitigate these effects and not really pay any of the expected real negative consequences of air exposure or hypoxia. So all in all, they seem to be well adapted to the challenges of their breeding environment, and that's changed the way we think about this parental care conflict with offspring. We kind of thought there was this system where the offspring wanted one thing, breed in the really hot, low oxygen intertidal zone. And the parents were like, no, that's hard, but I'll do it for you. I'll make that sacrifice. It seems like they're not paying as much of a sacrifice as maybe we initially thought, based on their various adaptations to dealing with these conditions. If you want to know a little bit more about that, my work with Segal is published in a journal called Physiological and Biochemical Zoology. Here's the link, and I can send that to my mom, and she can send that out. And there's also a Popular Science article that was written about that I'll also send out if you're interested. But that sort of concludes my first research story. I'm going to dwell really quickly on my transition into microbes. I'm not really going to talk about the work I did with Reese at University of Ottawa. But this is just briefly answering the question of why did I shift from the ocean to the lab? Why shift from fishes to microbes? And my concise answer to that is that microbes are really important and they evolve extremely quickly. Okay, so we already know microbes are important, obviously, because they can be pathogens, and that's bad, and we should study them because they're pathogens. I wanted to highlight a few things they do that's good for us. For example, cyanobacteria in the ocean produce about 50% of the oxygen we breathe. Yeast, like this Saccharomyces yeast, produce different products for us. The ethanol in the beer that we drink, or they help us make bread. I'm making bread later today, so thank you, yeast, microbes. And even though they are pathogens, they also can help us produce products like antibiotics that help us fight pathogens. So microbes are important. They also evolve extremely quickly. So here I've just put up the sort of generation time, or, put more simply, how long it takes some of these common study organisms to reproduce. Fruit flies, 2 days at max speed. That seems good. But in microbes like these bacteria, they can actually reproduce every 20 minutes if they're in the right conditions. This is so useful because it lets you bring microbes like these bacteria into the lab and passage them and come up with an evolutionary hypothesis. If we grow them in one environment for many generations or another environment for many generations, what do we expect to see in their evolution and their adaptation? And we can actually test these hypotheses by tracking evolution happening in real time. And that's called microbial experimental evolution, and it's an extremely important modern approach to answering evolutionary questions. I can't talk too much about that, but you can ask me questions about it later. But that's one of the main motivating factors for me switching from the ocean to the lab. If you want to read about some of the work I did with Reese, here's the name of the paper that we published together in the American Naturalist, and here's a link, and again, I'll send that out. Okay. Now I'm moving on to some of the work I'm doing at Yale, and I think I have 10 minutes. I think I can swing this. I might go a little bit over, but there'll still be time for questions, I promise. So this second sort of story is about using killer viruses to treat chronic bacterial infections. I'll just skip through the title slide. Okay, so I kind of want to put this question to you guys to have you guys take a stab at this, but since we're running low on time, I'll just do this myself. I thought I would highlight the differences between bacteria and viruses because it's often confounding, especially to third-year microbiology students who, even though we teach them what they are, they sometimes get it wrong on the exam. So I'm just highlighting a few important differences here. So bacteria are much more similar to animals. They are cellular organisms. They're made up of individual cells that sometimes clump together, but each of these is an individual bacterium clumped together. They reproduce kind of in a somewhat similar way to how most of our tissues reproduce, our cells within our body reproduce. It's called binary fission. It's basically the splitting of one mother cell into two clones, more or less. They're also capable of independent growth, by which I mean growth independent of a host. So independent of, we might have a E. coli that's from a human's gut, but we can actually grow it in the lab without any human's tissue involved in that at all, as long as we have the right soup of nutrients. You can grow them outside of a host. They're also, compared to viruses, this might seem small, but they're actually relatively large. An E. coli cell is about 1/1000th of a millimeter, which is pretty big as far as, compared. In the virus world, that would be huge, although there are some gigantic viruses. Viruses, on the other hand, are a completely different form of biological entity. Some would argue not alive. I might argue alive. And they're non-cellular. Basically, viruses are just made up of genetic material surrounded by proteins and sometimes fats that it's just a spaceship to get between host cell to the next host cell in order to infect it. And the only way they reproduce, they don't reproduce by binary fission where they split from one into the other. No. They always reproduce by getting their genome into a host cell, their genetic material, their genes, and that in effect reprograms that cell to become a factory for producing more copies of the virus, which usually then burst out of that cell, with dozens or even hundreds or thousands of copies of that virus, which can then go on to infect new cells. And they're relatively small. They're about 1/10,000th of a millimeter in length in some cases. Some are smaller, some are larger. Okay, so that's bacteria and viruses. For now, for the rest of the talk, we're going to focus on phages, which are, I'm not sure what the right plural is actually, which are viruses that specifically target and kill bacteria. Bacteriophages or phages, viruses that target bacteria. Here's basically how a phage life cycle works. We have this virus that's interacting now with a bacterial cell. That bacterial cell is covered, for simplicity, with just one type of protein that studs the outside of the cell, called the cell membrane. And the virus will specifically recognize one of these proteins, which is called its cell surface receptor, if you want the jargon. And it'll attach at that site, and then it'll inject in its genetic material, its genes. You can see here's the host genes, and here's the virus's genes. And those genes will be copied. It'll make many, many copies within the host. And the genes from that virus will also be used to reprogram the host into making many different proteins that those genomes are then packaged into all together. And eventually, you see the cell bursts, and we see many copies of the assembled virus burst out to then go and repeat the cycle and infect a new cell. So, needless to say, this kills the bacterial cell when they all burst out like that. And that raises the question of, can we use these viruses of bacteria to treat bacterial infections, similar to how we would normally treat them with chemical antibiotics like penicillin? I could tell you a lot about the history of this approach, which is called phage therapy. It's a lot older than you might think. It predates antibiotics, and we can talk about that a little bit later. But for now, I want to talk to you about how we can use evolutionary thinking to improve phage therapy. Okay, so here's my boss, Paul, and here's the longest standing researcher besides Paul in the lab. His name's Ben Chan. And here's sort of their idea for how to use evolutionary thinking to improve phage therapy. So they kind of asked, and this might not make sense now, but hopefully it'll make sense in a second. They asked, what if we could force bacteria, pathogens, specifically pathogenic bacteria, into an evolutionary catch-22? This scenario where they either die as the result of infection with phage, viral infection, or if they're able to resist the phage, if they're able to evolve to resist the phage, in doing so, they make themselves susceptible again to antibiotics like penicillin. Can we figure out a way to engineer this sort of situation where the bacteria are damned if they do, damned if they don't, in terms of susceptibility or resistance to these phage? And the basic approach to engineer the situation, it's not as complicated as you might think. All we need to do is find a phage that specifically targets and kills antibiotic-resistant bacteria. Maybe easier said than done, but just to explain a little bit more about the mechanism or the molecular details of how we might do that, we can go back to this sort of chart. And remember how I said that this bacteria cell is studded with these different proteins that the virus can recognize? The trick here is to find a virus that specifically recognizes proteins on the surface of the cell that allow that cell to resist antibiotics. And that way, that virus isn't going to just target any cell. It's only going to target the cells that are resistant to antibiotics, leaving behind the susceptible ones. And here's a little graphic of that and how that might work. So we might imagine somebody's infection. Somebody has a bacterial infection, maybe it's in their lungs or in a burn. And we see that most of the cells in this infection are resistant to antibiotics. They're the red type, which are resistant to antibiotics. Maybe we have one or a rare type that's susceptible to antibiotics. And then we add in this phage, in this case, a phage called OMK01, which has these properties that specifically targets and kills all the resistant bacteria, leaving behind only the susceptible bacteria, which are easier for doctors to manage because they're susceptible to antibiotics. If we apply the phage and the antibiotic at the same time, oftentimes in certain cases, our lab has been able to completely clear these infections, which are really, really difficult to otherwise clear. I'm running a little bit low on time, but I'm going to take you through one example briefly of where this worked, the first successful use of phage therapy using this approach in our lab. Here's a fair warning. There's an image on the next slide of an infected aortic graft. It's an open chest wound, or an open chest, surgically open chest. If you're really squeamish, this would be a good time to look away. I'm sure Penny, my aunt, is going to be fine with this as a former nurse, but others who might be squeamish, feel free to look away. Okay, here it is. It's scary. It's gross. But basically, there's this doctor, an ophthalmologist in Connecticut, who had to get an artificial aortic valve or aorta put in. Don't exactly know how this works. I'm not a heart surgeon. But as they were doing this, as often happens, bacteria, specifically a bacteria called Pseudomonas aeruginosa, caused a biofilm infection, caused an infection of this guy's heart. And this infection quickly became resistant to antibiotics. They couldn't treat it with antibiotics at all. And so this guy was going to die. There was a huge amount of inflammation, a huge amount of, side effects on this person's heart. And this person was pretty, was up there in years and was probably going to die because we lost the ability to control this infection. And so we use this approach where we use the phage and we use the antibiotics at the same time to have this synergy, this catch-22, placing the bacteria in the catch-22, and it worked. This person's chest was opened, the antibiotics and the phage were applied directly on top, and it saved the guy's life. The infection very quickly, within a few weeks, went away, and the person lived several more years after that. And I'm not sure if he's still alive, but he did not die of complications of this infection. So this was a huge advancement, and it sparked a lot of discussion about using this approach and really, in some ways, popularized phage therapy again in the US. Okay, so I'm just in the next just couple of minutes, I know I'm going to go a little bit over 11:50, but I'm going to talk a little bit about what I've been doing in the lab and the problems that I'm trying to solve using evolutionary thinking. So there's a bit of a problem with phage relative to antibiotics, and that's that phage, unlike a lot of chemical antibiotics, which, as you probably know, if you take antibiotics, sometimes it wipes out all the microbes living in your gut, and that can be really unpleasant. On the contrary, phages tend to be really highly specific. And even within a species, let's say this is a species, these are all members of the E. coli species, phage are usually only capable of infecting a small subset of different strains within a larger bacterial species. This is all well and good when you have an acute infection like the heart guy, because usually that's only one strain, one type causing that infection. But it becomes more complicated when you have a chronic bacterial infection. This is a kind of infection where the bacteria colonize some environment within your body, and they're able to live there, persist there, evolve there, diversify there for years or even decades. That might cause these populations to become more diverse, and it might cause there to be variation within that population and whether or not a certain virus can kill them or not. So that's kind of what we wanted to investigate in my project. And we wanted to ask whether phage susceptibility, which is whether or not the bacteria can be killed by a phage, can vary within these pathogenic bacterial populations, within these infections, for example, in somebody's lung. And whether if we look at different people, bacteria from different people's lungs, are we seeing a similar story all the time? Or are we seeing different stories? Or is each patient completely different with these chronic infections that have been evolving within their body for years or decades? Again, trying to be a little bit quicker here, but the system that we used was, again, it's another Pseudomonas aeruginosa example, same bacteria as the heart guy. But in this case, they're infections in the lungs of people with cystic fibrosis. Cystic fibrosis is a genetic condition which predisposes people, among other things, to have really harmful chronic bacterial infections in their lungs that are really, really hard to treat and cause a lot of death and harm in general. These infections are also well known for being pretty diverse. We worked with collaborators at Emory University to work with only four patients that had these types of infections. And instead of what is typical of collecting a single bacterium or maybe 2 or 3 bacterium from each of these patients from their lungs, instead we collected, in other words, 40 bacteria from each of these patients, giving us a total of 160 bacteria, 40 from each patient. And then within these were sent to us a couple of years ago now, and they asked us, hey, is there variation within these populations taken from a single patient or across 4 patients? But within each patient, is there variation in their susceptibility to the phage you use in the lab? And I'll just go over those results really quickly, and then we can break for questions. So just to orient you on this plot, on the y-axis, don't worry about the letters, but this basically means as you go up, a bacterium is more susceptible to a phage. As you go down, they're more resistant, and this dashed line means they're totally resistant to a phage. And these are 4 different phages that we use in the lab. And when we look at the first population we examined, we immediately were shocked. We said, wow, there is so much variation within these populations. About half the bacteria are totally resistant to all the phage that we have in the lab to throw at it. And about half the population are susceptible, but even those vary. If you look at, in response to LPS5, half are resistant, and the half that are susceptible vary from being really, really susceptible all the way down to being barely susceptible. That's what this graph is showing. And there's a different pattern across the different phages. Now let's look at another patient. We see a pretty different pattern here. Most of them are totally resistant. And again, we see a lot of variation where they are susceptible. Now let's look at another patient. A different pattern. Again, tons of variation in whether these bacteria are susceptible or resistant. Let's look at another patient. Wow, a totally different story here. All the isolates within this population, all the bacteria were really susceptible, which is good news for this patient, to the viruses we study in the lab. What does this mean? It means that in these chronic infections, we can't take for granted that if we take one bacteria from that patient and another bacteria from that patient, that they're going to be similar and that we can use a similar virus to treat them. That's a problem because oftentimes we're just, the conventional approach to maybe antibiotics in some cases, at least historically, has been you have this kind of bacterial infection, here's the antibiotic. But in reality, we need to treat these patients as individuals and figure out not just, what one bacteria we sample from their infections, how we can kill that bacterium, but really we probably need to sample multiple bacteria from their infections to try to figure out how many different types of viruses and antibiotics in combination do we need to use to clear these infections. And each patient needs to be treated differently. Okay, to sum that up, I just want to leave you with one final slide, coming back to this idea of evolutionary thinking and seeing biological problems through the lens of evolution. So I hope I've convinced you, I think I have this written down, that I hope I've convinced you that evolutionary thinking can be a useful approach for tackling some of the problems we face, big and small, big like antibiotic resistance or HIV vaccine conspiracies, and maybe small, more like, can this fish deal with living in the intertidal zone? And then I wanted to leave you, similar to some comments that Jacqueline made last week. Evolutionary thinking is just one of many useful approaches. And we need collaboration between evolutionary biologists and other researchers and beyond researchers, politicians, sociologists, members of the community to help solve these problems. Okay, that's all I've got. Thank you so much for listening. And I know I went a little bit longer than I probably should have, but I'm happy to take any questions.
Beautiful. You are not a biotech medical researcher. I like your adjustment of your Instagram.
Well, which do you like?
I'm going to open this up to our community who has some questions or comments that they want to ask Noah. Just unmute. Make yourself known. Mark.
Very thoughtful and enjoyable presentation, Noah. I really enjoyed your slides. A couple of historical things to think about. I mean, I could ask lots of questions, but Enid I don't want to hog she comes second. First, there's you mentioned, you tweaked my memory when you showed the slide of the polio story and HIV and sub-Saharan Africa. There's a couple of really nice examples of evolutionary biology similar to that. There's, as you know, and you also showed a slide on the energy metabolism. There's an important enzyme, G6PD, to metabolize sugar in, I forget whether it's the aerobic or anaerobic cycle. I can't remember that. But some black people, or black people are more commonly deficient in this enzyme. But it was found that in Africa, if you are deficient in this enzyme, you're more likely to be resistant to malaria. So again, evolutionary wise, it was the malaria would wipe out people in populations, but certain black people were resistant to it. Now, there's a downside of that story. In the Second World War, when Black Americans were fighting in the South Pacific, they were given, anti-malaria pills, quinine pills, which stress the oxidative system. And oh boy, the Black soldiers taking these pills got terrible anemia. They got hemolytic anemia and they ran into trouble. So there's an evolutionary biology story, I think, going both ways, pro and con.
Yeah, no, absolutely. Yeah, no, the malaria story and especially as it relates to sickle cell is a fundamental example in evolutionary biology, and we learn about it a lot. And I think the evolutionary thinking is extremely useful here because it can help us solve like these extremely curious situations. Why are there these mutations in genes that are so important to survival persisting in populations? Why? Why do so many people in these areas of the world where malaria is prevalent have these mutations? It's so hard to explain until let's use evolutionary thinking and think, well, these people have had to deal with the challenge of malaria, and having one copy of this mutation gives you really high resistance to malaria. And so over many generations, that can be favored. We can't really understand that without considering disease ecology and evolutionary biology, I think. And so it's so useful, I think, to, if they were using evolutionary thinking, I don't know that quinine would be administered, unfortunately, to those soldiers.
I have another question, but I'll let other people go, and then I'll have something for you to think about.
Okay, Penny, did you have a question?
I just want to know if you can send me a phage or something to get rid of my cough.
Well, that's a good question. I don't know. I need to figure out what's causing your cough, Penny. Is it caused by bacteria or a virus?
I diagnosed myself with bronchitis.
With bronchitis. Okay, very good. Probably not. Probably can't. But you know what? If you had a little lab set up there, I promise you, phages. I should have said this before. Phage are the most abundant biological entity, is what we like to call them, on Earth. So there's about 10, this is the estimate, 10 to the 31 phage on Earth, which is 1,000,000,000,000,000,000,000,000,31 times. That's how many of these viruses are on Earth. So they are all over your skin. They're in your gut. They're in your sink. They're in the soil. They're everywhere. So you could go find one for yourself, I think, Penny.
Okay, I'll try.
Okay, good luck.
Thank you.
You're welcome.
Enid. From my lab training, I remember, though it wasn't microbiology wasn't my specialty, but I remember when we cultured sputum and grew Pseudomonas at colonies, they had a particularly fruity odor to them. And also, as you mentioned, really hard to get rid of in the lungs. I think if I'm correct, they use quinolones to antibiotics to try and get rid of Pseudomonas, which I know my sister-in-law has that issue and they keep switching antibiotics on her. With all the stuff that's going around and that Penny has, and I've had, we get into the overuse of antibiotics. I don't know whose fault that is because I know patients bug their doctors that they want to get better and they want to get better right now. Give me an antibiotic. We also have a lot of antibiotic resistance in hospitals. Nosocomial, which means hospital-acquired infections. I don't know what the answer is to that, because whatever we develop, is there the same danger that we're going to overuse it and people become resistant?
Yeah, no, absolutely. It's a massive problem. I can say as somebody, and you're well aware of this, but maybe others aren't, there are so many brilliant people working on this problem of rising antibiotic resistance levels. And this has been a problem we've been aware of for some time. And it's due to the overuse of antibiotics. By the way, not just in medical contexts. It's not the only context where antibiotics are used. A huge driver of the antibiotic resistance crisis is the use of antibiotics in farm animals, and it can help to fatten animals up, make them grow up to be healthy and big. I don't know. We can ask Penny if they use antibiotics on their on their cows. No, okay, that's good. Antibiotic free. That's good. That's a huge part of the problem too. And there's been a lot of policy changes. I think there's certainly less antibiotic use in farm animals now, and it's used more judiciously in hospitals. But antibiotic resistance levels continue to be really high. And the solution to that, there's probably a lot of them. You know, I can selfishly say I think more investment in academic research, not less, is probably good on that front because, as probably a lot of you know, one reason why antibiotic resistance is a huge problem is because antibiotic development has been stalled. Because why on earth would you spend billions of dollars developing a drug that the bacteria are going to quickly evolve resistance to? It's not economically viable. But it's a little bit more viable for academic researchers who don't have to emphasize profit to look for things like phage, right? Which are, by the way, pretty cheap to find. I could go out right now today, take some soil, put it on some bacteria, and find a new phage and try to characterize it. But my point is that academic researchers have different incentives, and we can explore a diversity of approaches, and that just doesn't just mean finding new antibiotics. It can also mean finding new approaches to how to manage antibiotics too. And if you read some of my master's supervisor, Reese Cassin's work, he's thought a lot about the optimal way to use antibiotics that'll minimize in human communities and human infections, antibiotic resistance. But thank you for your comment. Oh, yes, there's one over here.
Thank you for an excellent talk and great slides. I wanted to ask, I'm Hinda, by the way, seeing as
Hello.
my name on the the question I wanted to ask is about AIDS, and you had said that it's believed that the AIDS virus had been around for decades prior to I guess the outbreak or the HIV virus. Can you say a little bit more about that?
Yeah, absolutely. So spillover is kind of a fascinating concept. Spillover is what happens when an animal species that is usually the one that houses the virus. Oftentimes animals can deal with certain viruses asymptomatically, and they can circulate in animal populations for thousands and thousands, millions of years. And then they can spill over into humans and cause quite a big problem, as HIV has around the world and continues to be a huge problem in certain areas of the world. So what is thought to have happened with the origin of HIV, of course, we don't have a great idea because we don't have the samples to track when it first spilled over, but the sort of leading theory based on how long ago HIV entered human populations and sort of the geographical distribution of different sequences and their diversity, what probably happened was there was a single person who was maybe, you know, one common theory is that they were hunting for bushmeat, which is to say they were looking for food in the forest. This would be like late 1800s, maybe the 1880s. You know, Canada's just become a country at this point. And they're hunting in the forest and they kill a chimpanzee that was infected with simian immunodeficiency virus, a close cousin of HIV. And during the slaughtering or the butchering of this animal or while eating it, they expose themselves maybe through a cut on in their mouth or maybe a cut on their skin to this virus. The virus can then circulate at really, really low levels for many, many decades, even close to a century, without becoming a huge, massive problem. You know, can circulate in relatively few individuals. And the thought is that maybe this person infected one other person 5 years later. And then that person maybe infected 2 other people 10 years later over the next 10 years. And slowly but surely, it's thought that it made its way into different cities in Africa, like Brazzaville, I think is the name of one city, which is thought to be an early epicenter of the sort of AIDS pandemic. And it can happen unnoticed too. Somebody's getting sick, somebody's dying in one of these contexts where there's maybe not as much medical surveillance as there was in North America. It could be anything. We don't know that this is a new pathogen that's causing this all the time. And that's how a virus can go undetected, circulating in human populations, increasing in size until it reaches a place like North America, where all of a sudden people, relatively healthy young men, are getting sick, and they're getting these crazy diseases where they're getting flush, you know, where fungal infections are causing mouth infections, which are never happening in healthy people. Doctors are saying, what's up with that? And they have the resources to investigate and figure out what's behind it. And then lo and behold, we find a virus. So I think the answer to your question is that it can circulate undetected for a long period of time if it's at low frequency and it's in an area of the world where the medical surveillance at that time was not the same level as it is today, especially North America.
So what you're suggesting is it wasn't as though this, it was an abrupt outbreak. It had been there, it was a gradual and it just was undetected.
Exactly.
Okay, thank
Yeah.
you.
No, I have a question for you and Jacqueline.
Okay. Oh, nice.
Combined evolution, anthropology, and I don't have the answer, but it's something for you to think about. If we go back, it's about the Jewish people and the BRCA gene, the breast cancer gene, was isolated on chromosome number 7. We know it's common knowledge that the BRCA gene is much more common in Jewish people. Now, think about it. Want to go back to the Bible, but go back to the Middle Ages, etc. Jewish people lived together. And there was a lot of marriage of cousins and all that sort of stuff. And so this mutation that became more prevalent. Fast forward to gets into the survival of the fittest and the adaptation, different kind of adaptation. 1492, 1493, this in Spain, where Jews had been very successful and so on and so forth. There's the Inquisition. Jews are told to convert, and many are killed, etc., etc. Some are able to migrate successfully to North America over time, and the Jews or the people that converted Moranos settled in many of them settled in southwest or either Southwest United States, Mexico, and you see them doing some funny. Fast forward years and years and years, generation to generation. They still light candles on Friday night. They're Catholic. They light candles Friday night. On their gravestones, you see Star of David, stuff like that. Shin, that sort of thing. And when they've done what Jacqueline was talking about, anthropologic studies, but studying the genes, they see, gee whiz, these people have a lot of BRCA gene. So it's kind of an example of how evolution, if you will, survival of the fittest, they converted or they practiced quietly, but they left. I mean, that's sort of a behavioral change, not your classic biologic thing, but it is, to me, fits into what you're talking about. I had a little pet project here where the small Portuguese population in Hamilton, very inbred, but they all come from the Azores. I would see breast cancer like every generation, grandmother, mother, daughters, sisters, and my hypothesis was the Azores were islands off of Portugal, and in 1493, they did the Inquisition in Portugal. A whole bunch of Jews or who weren't killed, practicing, quietly, who had Moranos, etc., went to the Azores. Eventually, fast forward many years, the folks from the Azores migrated, some of them to Hamilton. Again, very inbred. High concentration of the BRCA gene. So again, I wish more people in the cancer field, cancer research, were thinking of evolutionary biology as opposed to, or bringing molecular biology and evolution and migration together to think about why there's many unanswered questions.
Yeah, Jacqueline, do you want to take something from that first? Okay. You said a lot, Mark, and that's a fascinating story. I'll just say one thing that I picked up on in what you said, which was the idea of a cultural change, hiding one's religious practices, right, in order to survive. Survival of the fittest in that way. It's not the big fish that are getting killed in that case. It's the Jewish people, who are getting killed. Adapt or die, I guess. And there's a cultural change that leads to higher survival in people who sort of hid their religion. That's something that we study too, which is pretty cool. There's a lot of people evolutionary biologists who study stuff like cultural evolution, and you can see it happening in human populations. You can see an interaction, in the lactase persistence example I gave you, between a cultural change, farming animals that produce milk, and then a molecular change, the persistence of the lactase enzyme through adulthood, which allows you to process milk. So it's fascinating trying to understand cultural evolution, how it maybe interacts with more traditional genetic evolution and stuff like that. It's something that a lot of people study as well. So yeah, thank you for the comment.
Wonderful. We're going to thank you so much, Noah, for this presentation. Every time I hear you talk about it, I learn new things and understand a little bit better what you're doing there in the lab at Yale. So thanks very much for being with us today. 2 weeks from now, on February 23rd, we'll hear from Renan Dahan about his work in engineering and in particular 3D printing and why he has found that so interesting for little inventions and things that he's doing. So very exciting. Wish everyone a beautiful afternoon here at the temple where I am. This afternoon we have a Purim rehearsal, and this evening we have a continuing series called Jews in Space. We're looking at clips and discussing things like Star Trek and The Empire Strikes Back and so on, and where there are similarities with our Jewish history, culture, and religion embedded in those stories. So interesting. Interesting.
Okay. Thanks, everybody. Nice talking to you.
Thank you.
Thank you.
Thank you.